A systematic review is conducted by carrying out a comprehensive search of published literature using a pre-specified protocol and summarizing the studies identified. A meta-analysis can be conducted in order to pool the results of individual studies to create a quantitative summary of their results. There are several different types of meta-analysis, with individual patient data meta-analyses considered the gold standard. There are several reasons for carrying out a meta-analysis. For example, to increase statistical power, which means the chance of detecting a statistically significant effect, if it exists, to improve precision, which leads to a more accurate estimate of the benefit of a particular intervention, to investigate questions not posed in individual trials, and also to compare treatments where no head-to-head trial data are available. However, a meta-analysis should only be carried out if studies are similar in their characteristics. For example, patient population, study design, and reported outcomes. If the studies included in a meta-analysis are clinically or methodologically diverse, the results could be meaningless and final conclusions misleading. Meta-analyses must be undertaken with care and should follow a pre-specified protocol that outlines assumptions and methodologies to be applied. This will ensure that any conclusions made are drawn on the best available evidence. There are 5 areas that should be considered when planning, conducting, and reporting the results of a meta-analysis. These are the data to be combined, the measures of effect between studies, the differences between studies, how to deal with potential bias, and how to determine the robustness of the findings. We will look at each of these in turn. First, let us focus on the types of data that can be included. The 3 types of data that are most commonly combined in meta-analyses are dichotomous, continuous, and survival data. There are a number of statistical techniques available to analyze each type. With dichotomous or binary data, a subject is in one of 2 states, dead or alive. These data can be summarized using odds ratios, risk ratios, or risk differences. Continuous data are those such as age or functional assessment scales. These data are summarized as standardized differences in means. Finally, there is survival, or time to event data. These are commonly used in oncology trials where the principal outcome is the time to the occurrence of an event, for example, progression-free survival. These data are summarized using hazard ratios. A meta-analysis does not simply involve pooling the data to calculate the arithmetic mean. Instead, a weighted average is calculated taking account of individual study characteristics such as study size, patient characteristics, or the number of observed events. Several statistical techniques are available to assign weightings to individual studies. The choice of these depends on several factors, including the type of data combined and an assessment of heterogeneity across studies. A common way of displaying both the weighting assigned to individual studies and the results of a meta-analysis is a forest plot. The results of each individual study are shown as boxes. The box sizes are proportional to the study weight, and the horizontal lines represent the confidence intervals. The weighted estimate across all studies is shown at the bottom of the plot as a diamond. Now let's focus on how measures of effect between studies are calculated. Generating a summary measure of effect from more than one study is one of the main goals of a meta-analysis. The 3 most commonly reported effect measures are relative risk, odds ratios, and hazard ratios. Relative risk refers to the probability of an event with the use of a particular intervention compared to the probability of the same event occurring in a control group. It is calculated by dividing the risk of an event with an intervention with that of a control. In this example, the relative risk is 0.5. The intervention can therefore be said to be associated with a 50% reduction in the risk of an event. The odds ratio is calculated by dividing the odds of an event occurring with an intervention with that of a control. In this example, the odds ratio is 0.44. Interpreting an odds ratio is slightly different than for a relative risk. Therefore, it is sometimes easier to convert the odds ratio into a relative risk to aid interpretation. Hazard ratios are used to report relative risks per unit of time. They are similar to risk ratios, but differ in that they measure risk at a specific time point, and may change over time, for example due to age. Let's now look at how differences between individual studies are assessed. An essential part of a meta-analysis is the assessment of between-study heterogeneity to determine whether individual studies are similar enough for their results to be combined. If the study characteristics, methodology, or reporting are too different, the data may not be suitable for meta-analysis, and carrying out an analysis could lead to erroneous conclusions or misleading results. There are three main areas where differences between studies may be observed: clinical diversity, methodological diversity, and statistical heterogeneity. Clinical diversity refers to differences in participants, interventions, and outcomes measured. Methodological diversity refers to differences in how the studies were conducted. For example, in study design, study quality, or in the methods used for randomization and analysis. Finally, statistical heterogeneity refers to whether the results of individual studies vary more than would be expected by chance alone. Clinical and methodological diversity can be assessed by comparing summary data tables from the systematic review on which the proposed meta-analysis is based. However, statistical heterogeneity is different. One way to assess this is to view a forest plot of the individual studies. When the confidence intervals do not overlap, there is likely to be more variation between study results than would be expected by chance. Alternatively, a chi-square test can be run to measure whether observed differences in study results can be explained by chance alone. However, this test can have low statistical power if there are few studies. If heterogeneity is suspected after these tests are run, it is important to consider whether it is still appropriate to conduct the meta-analysis. It may be better to only qualitatively summarize the data. If meta-analysis is deemed appropriate, then subgroup analyses and metaregression can be carried out to take account of heterogeneity, remembering that these are essentially observational in nature as they remove the benefits of randomization in the individual trials. As such, random effects models may be used. For example, when carrying out a meta-analysis, a fixed effects model can be used to estimate the impact of a new intervention as if there were a single true value underlying all results. Any variation seen would be assumed to be due to chance. However, when heterogeneity across studies is suspected, a random effects model is preferred. A random effects model is different in that it assumes that there is no single underlying value of effectiveness, but a distribution of values depending on study characteristics. Random error and between-study variability are incorporated to calculate a more conservative estimate of results than fixed effects analysis. It is good practice to analyze data using both effects models and report the results that best fit the data. Let us now focus on how to identify potential bias within a meta-analysis. Systematic reviews, and by extension meta-analyses, aim to identify all published literature on a given topic. However, reporting bias means that any analysis is unlikely to identify all possible data on a given topic. This is because studies that show a significant effect and/or positive results are more likely to be published. This may mean that the meta-analysis is only based on a subset of the potential data. This could lead to the overall summary effect being exaggerated. There are many different types of reporting bias which can affect the final results of a meta-analysis. We will now look at publication bias in more detail. Identifying publication bias can be difficult as unpublished studies cannot be accounted for. A common method of assessing publication bias is a funnel plot. This plots the individual study effect estimates against a measure of the study's precision or size. Effect estimates from smaller or less precise studies tend to be scattered more widely about the pooled effect estimate, while larger or more precise studies are usually closer to the pooled estimate. If there is no publication bias, then the graph should resemble a symmetrical funnel. However, if smaller studies have not been published or included in the analysis, then the funnel plot may not be symmetrical, indicating the existence of publication bias. Occasionally, certain factors, such as per-patient inclusion criteria, may affect the likelihood of a particular study estimating the true effect, which could lead to there being outliers on the funnel plot. Finally, let's look at how the robustness of the findings of a meta-analysis is determined. To determine the robustness of a meta-analysis, a sensitivity analysis can be performed. This will answer the question: do the decisions made in the analysis impact the strength of the findings? If the answer to this question is no, then the results of the analysis can be viewed with a higher degree of certainty. Decisions taken at each stage of a meta-analysis may affect the final results. For example, the sensitivity analysis may need to consider what impact the inclusion of data only published as abstracts has had on the analysis, or whether the results would be different if a random effects model was used instead of a fixed effects model, or vice versa. In this video, we have covered what a meta-analysis is used for and the factors that must be considered when conducting one. We have discussed how it is important to be familiar with different types of data and measures of effect, how to deal with heterogeneity between studies, potential bias, and the role of sensitivity analysis in determining the robustness of the final conclusions.